All right, so that was the final talk of the morning session, but we have a roundtable that's coming up. So I would like to invite to the roundtable Wendy, Mike, Jillian, and Julie.
Yeah.
So for this roundtable, I thought we would focus on biomarkers. Surprise, surprise, probably guessed that by who I have sitting up here. And so, I mean, I will just start off with a question and that I'll make everyone answer, but we won't hold you to your answer. But when are we going to have a biomarker that's specific for STXBP1 that can be used in the clinic?
On Monday. I don't know. Molecular biomarkers are incredibly difficult. I think some of the physiological biomarkers that were presented today have a really good chance of being in the clinic.
Yeah, I'd say very soon, or we already do. Just need to be, I would like to validate them in more settings, but I think we are close for electrophys.
I think the sensor data is much further away. I think we would need, I want to be at the point where it's so clear that it is a biomarker that it's like not disputable. You know, I feel like we need really, really strong data, longitudinal data, and responsiveness. There's just a lot of things that are missing. So my data is much more preliminary, but I hope that all this happens in the next few years.
I'd like to reframe that and say biomarkers, because I don't think that there's only going to be one biomarker. I think we've all given very different talks and taken a view of biomarkers in a different way, and I think there needs to be more than one. From a molecular point of view, it would be great if we have a few molecular biomarkers. Then there's also EEG markers. There need to be a few, more than one.
Okay.
Completely agree with that, yeah.
And so to follow that up, when I had asked the question, I said specific to STXBP1, but do we need a biomarker that is specific to STXBP1, or we just need a biomarker that is going to show a difference between, with treatment? And it could potentially work for any, let's say, NDD or DEE.
I don't think it needs to be specific to STX as long as it's responsive and tracks with outcomes.
I totally agree. With STX, it's a genetic diagnosis. You have a pathogenic variant you diagnose. Unlike other disorders like Rett syndrome, where it's a clinical diagnosis where you need something pretty specific. I think for STX, I don't think it needs to be specific to a disorder.
I think I would like to see a specific STXBP1 biomarker as one of the several biomarkers.
You're thinking of target engagement, yes?
Yes.
So does anybody, again, this is open up to everybody who is in the audience to discuss this. And so how do you feel about, you know, based upon what you've heard, maybe you have an idea that, yes, you think that we'll have a biomarker in the next year or 2 years, or that, yeah, you should have something specific, or both, or whatever, yeah.
I want to add something about the biomarker, that not that what biomarker you are looking for, but where you are looking for. For example, and for what reason. For example, if you do gene therapy, so where you want to look at to find a biomarker. I make a simple example. We have creatine deficiency. We give patient creatine. We do MRS, and we see that creatine in MRS goes up, right? So looking at this, I know it's not that easy, but for example, if you do gene therapy, you want to see, okay, I saw a beautiful picture about mRNA that was there after gene therapy, but can we have biomarker to see in the brain, like STXBP1, bind with some kind of radioactivity or something that can be tractable? I mean, I know it could be science fiction right now, but I mean, it's like, that's the way I look at the biomarker. Thank you.
I want to add maybe a little bit to the discussion whether we need a specific biomarker or not. I think it really depends on what type of biomarker we're talking about. Of course, if we're following treatment effect, it doesn't need to be specific. But I think still we could use diagnostic biomarkers also. We had a lot of talks about fusses yesterday. If we have a disease signature or a diagnostic biomarker, that could really help interpretation there as well, I think.
Just wanted to ask, I guess, a pretty direct question. So it feels like the biomarker efforts are really on a great track. If you were to just project 3 years in the future, what do you think we're going to be choosing as like the primary outcome, secondary outcome, based on what you think you know today and the trajectory?
As a pharmacodynamic biomarker that could predict therapeutic efficacy, STXBP1 levels, potentially, certainly electrophysiology, and I really do think that the biosensor work. I really like the idea of this. I'm coming around to this idea of immune-mediated dysfunction in neurodevelopmental disorders that you can accurately see and assess in the periphery. And it really kind of brought it home in that combined brain symposium where disorder after disorder after disorder after disorder after disorder was seeing immune dysregulation. And we see some of that in STXBP1 as well. So I think that, it's really hard to predict the future. I would love to have a very specific molecular biomarker that we could hang our hat on to predict target engagement. And for therapeutic efficacy, we have a number of different, very promising clinical candidates. Whether that's going to be a molecular biomarker or not, I don't know. But I think with the outcome measures and the developmental milestone work that we're learning from the natural history study, as well as what we're doing with QEEG and wearable biosensors, I think we're well on our way to identifying meaningful outcome measures and endpoints.
I'd like to add to that. I think it's very, I agree with Mike, it's very difficult to predict what's happening, but looking at biomarkers and different biomarkers at a greater rate than we've ever done before. Thinking about other disorders like Rett syndrome, which is one of the older disorders, MECP2, the pathogenic variant of MECP2 causing Rett was discovered in 1999. And up until now, there have been more than 70 clinical trials that have been run for Rett, and only one therapeutic has been approved, and that was trofinetide 2 years ago. And a lot of the clinical trials were predominated, with the outcome measures were predominated by CARA and clinical report outcome measures. I'm not saying that these aren't good, these are very, very valuable, but there needs to be some other way of identifying how effective treatments are in clinical trials, because we, STXBP1, don't want to have 70 failed trials because the outcome measures, or there weren't biomarkers that could measure changes, either very small changes or measure changes fast enough before the clinical trials stopped. So I think it's really important that we move to these biomarkers and include them in the trial design so that we don't have any more failed trials.
Yeah, and to piggyback on that, Jan, you're absolutely right. The biomarker research and progress has been astounding in the last couple of years, and I think it will be even more astounding in the next couple of years. But it's important to note that none of the ongoing gene therapy trials in other neurodevelopmental disorders have quantifiable molecular biomarkers that they can benchmark their therapy against. None of them. Rett, Angelman, none of them have molecular biomarkers. So what we're doing here is actually, I think it's pretty awesome.
I wanted to add, I agree. I think this is actually an area where I feel really optimistic in that everyone who has talked today is measuring very different aspects of the child, and we're going to look more at the clinical data later, but we're capturing so many different areas through the EEG, through the sensors, through the molecular stuff, through the blood and the plasma, all the things that I don't do. But there's really a lot that everyone is doing, and so together I'm very hopeful that we're going to find the different pieces, and hopefully we'll see change across multiple of them.
So I have a kind of a comment that builds on what was just discussed, and it's a little crystal ballish, but there is all of this progress that's happening that I think is really exciting from like a research community and a family perspective. Being able to take this forward into clinical trials requires acceptance from regulatory bodies around, the utilization of these biomarkers. And the FDA, and the EMA have been slow around, acceptance of like sensor and motor type of endpoints. It took 10 years for the Actimio to be like actually accepted as a secondary endpoint in the EMA. It's not yet accepted in the US at that level. And so what can we do as a community to get the, ensure that we're doing, the generating the evidence and then also the regulatory engagement? Like for instance, I am extremely optimistic about the science in terms of the EEG data because we're starting to see this correlation with clinical data, and that's what the FDA cares about, right? It's how does the patient feel, function, and survive. But the FDA has also expressed a lot of skepticism around the EEG. So what
It depends
can we
on
do?
which FDA you talk to and which day, right?
But yes,
Michelle
but-
Campbell in an informal interaction said the biosensor data looks like an endpoint.
Absolutely, but
Right.
absolutely, but like the what we hear more broadly is, and I love Michelle, and I wish
Yeah.
that she was.
I wish she was here. Yeah.
Right, I wish she was the
Yeah.
FDA, but she's
Yeah.
not the FDA. There's
Right.
a lot more skepticism that you hear more broadly. So, what do we need to do, to create the right evidence and the right engagement?
I think that's what we're doing, is we're really hammering down on reproducible disease or disorder specific biomarkers that we can then take to the FDA and say, look at this evidence. In our CPIM meeting a couple of years ago, they were very skeptical about the EEG data. And they said, well, you need to be able to correlate with that with developmental outcomes. And Ingo said, we have that. And they went, are you kidding me? Their jaws hit the ground. So we're really pushing the boundary for biomarker identification and development in neurodevelopmental disorders within STXBP1 and more broadly.
And so Charlene kind of anticipated my point that I wanted to make. I think what we're seeing with the biomarker part is really this shift from discovery to validation.
Right.
Yeah, and I think this is probably, two things I wanted to kind of note. First of all, this came from nothing to where you guys are now. Yeah, so there's actually quite a big change that we can have biomarkers in different domains and assess this. And I think what we're seeing now is this change from there is something that we can track that looks very interesting to start with, to saying how robust is this? How can we what is the real signal there? What is just noise?
Yeah, it'll be very different and easier to write grants when you have biomarkers that you're now validating instead of trying to discover.
Yeah, and I think this is just like a frame shift that will get us into like the regulatory mindset where you have to really work on like, what are the concepts? How do we validate this? How can we show stability, minimal differences and things? So.
We need patients in the community to keep engaged with biosample collection, not only patients, but sibling controls where possible. I mean, we can't do any of this work without the patients. We absolutely have to have patients and controls to do this work. This isn't a mouse model. We can't just go to a mouse model and do this. We can learn some things from mouse models, but to actually develop a biomarker for a human condition, we need patient families and the advocacy groups to really work together with us.
Yeah, Yeah, and that's certainly already happening in our clinic. We see that every day. The families
Yeah,
are so
it's
committed
happening in
to
STXBP1.
getting the data.
So one question that comes up, and it was discussed, Mike, you discussed this, and others have discussed this, especially with the blood biomarkers, which is, so again, a lot of the therapies are targeting the brain. We are kind of hoping or assuming that what we see in the blood is what's happening in the brain, but, you know, how realistic is that? Now, we think that you brought up it may be neuronal exosomes or something. How do we determine that? Is there a way that we can determine that, or are we just going to end up, or do we have to actually wait until we actually are in a position where we've treated people, they've shown good improvement, and yet the blood levels of STXBP1 don't change?
So we don't know the answer to that question. I think that question's been asked about 4 times today, and we really don't know the answer to that. But we can gain some insight from the mouse models, doing experiments that we're planning to do and that BioMarin has already done and presented on in Heidelberg. And so the answer to that question is we do not know, but we will hopefully in the next year to 18 months.
We asked that question a couple of years ago with our RET biomarker study, and we were funded to look at omic analysis in brain organoids from individuals with Rett syndrome. So we grew up all the organoids to 180 days and did the full omics on them. And we're looking at that data now, and then we're going to compare, this is what we promised the NHMRC, which is our funding body, that we would compare the omics of the organoid data to that of the blood from the children that are described today, and we're hoping that we will see something that's similar. I know it's not a human brain, it's just a cortical organoid, but it might give us some indication of what we see in the organoids compared to the blood of the patients.
And so it's getting close to lunchtime, and I already hear some people's stomachs starting to rumble here, but I wanted to also ask a question, and Wendy, especially about what you presented with the multi-omics kind of biomarker discovery. And so, you know, this is something that is pretty popular. You know, Combined Brain has been running a multi-omics analysis and stuff. And so with that huge amount of data that you get from all of that, you know, how's the best way to analyze that data? What do we think that data is going to show us? You know, obviously you're running that, you're not being specific for STXBP1 or even specific for, you know, Syntaxin, but you have all of this data again. So how do you look at that? What kind of conclusions are you hoping to draw from that, from all of that data?
It's terrifying. The amount of data we have is absolutely terrifying. We have 4 bioinformaticians that work on the biomarker project. And also, I was so petrified of having something wrong with our code that we actually asked some folk at TGen to validate it independently. So they didn't look at the data, they just looked at the code, so that we're happy with our code. But the coding is one thing. The amount of data we have is insurmountable. We're swimming in it. And it is a lot of data. And what I presented today was just our take of what we think is happening. But there could be so many more things that are happening. And so that's why I'm quite happy to chat to whoever is interested in looking at this data, to validate it, to kick the tires, to see if it actually can hold up, and if there are any other ways in which we can look at it to find out more about what's happening.
Yeah, it's an incredible amount of data and an incredible amount of work. But the N is 22 patients. And I think we need to generate about 5 to 10 times the amount of that data to get a meaningful signal across multiple different omic platforms. Ultimately, it comes down to a numbers game at some point.
Absolutely. But if we take a coordinated approach to not only look for, this is what you said initially, Mike, not only look for biomarkers that can help us detect changes in disease-modifying therapies and early changes as well, but also targets as well, drug targets. Those, we need those as well. So gene therapies are wonderful, but it's great if we could have some small molecules. I think Bev mentioned that we could have as well. So this will allow us to, it's so powerful, the data, but I think it's going to take a coordinated approach to be able to mine it properly.
Does anybody, Sarah, are you getting up to ask a question or to go to lunch?
I was trying to decide about. I'm just kidding. To ask a question. So we talked a lot about using each of the biomarkers that you all are working on as therapeutic endpoints. But what I also thought was interesting in each of your talks is maybe the novel insights it gives into just understanding STXBP1 as a disease. And it's kind of an open-ended question, but I wonder if anyone would want to comment on it. Thank you.
Yeah, yesterday I disparaged basic science and was reprimanded for it, and rightfully so. I think that anytime we have an ability to learn about the underlying pathophysiology or molecular function while we're trying to develop something to help kids, it's a win-win, right?
Yeah, I mean, I think Melissa said it best this morning when she said we have the opportunity to stop searching and start learning. I mean, that's what we're still doing, right? We're still learning about these patients and trying to provide some answers about the phenotype. So we're going to keep trying.
That's why it's called research and not search.
Go ahead, Scott.
I was just going to make one comment on the wearables, which is some ways of analyzing that probably functions like a biomarker, but in some ways you're also just measuring functional outcomes. So it actually could be classified as an outcome or a biomarker, and there's probably some important strategy about which one you do in terms of how you're selling that to regulatory agencies. In either case, you're going to want to validate that it correlates to some other outcome measure that's relevant for the way you're looking at it. But it may actually, like biomarker, you have to kind of do extra jumps to say this means something that actually matters. If you're actually just measuring a functional component of what's going on with a person, that's that, right? And so it may be a great, when Michelle says that looks like an endpoint, she doesn't necessarily mean that looks like a biomarker endpoint.
Yeah, that's exactly the point. I said, what do you think of this as a biomarker? What do you think of this data? And she goes, it looks like an endpoint. Yeah, you're absolutely right.
All right, well, if there are no more comments or questions,